This video will cover solutions to the 2017 ENSA Section 2 paper. Just as an editor's note, throughout the video, references will be made to the 2016 paper, but rest assured that this video will cover the solutions for the 2017 paper. This video will cover question B1 from the 2016 Section 2 of the ENSA paper. So, in part A, we're given two figures of cells, and we're asked to work out what type of cells we're looking at. So, as ever, when presented with a light or electron micrograph, we should work out whether we're looking at a eukaryotic or prokaryotic cell, namely an animal or plant cell, or a bacterium. So, what we should be working out when looking at our eukaryotic cell, if there's one present, is whether it's a plant or animal cell, since we're asked for a specific type. So, we're looking at part 1 here, and part 2 here. We can see that the basic structure which is visible is a cell wall in both cases. Well, these can be found in plant cells, but also in bacteria, which are prokaryotic. So, we should look for other structures, such as the presence of membrane-bound organelles, which can be found in figure 1. In this case, we can see it is a nucleus, since it contains a smaller nucleolus within it, and it's particularly large and dominant within the cell. There are a number of other membrane-bound organelles, which are only present in eukaryotic cells. Moving on to figure 2, there's only cytoplasm and no membrane-bound organelles within the cytoplasm, meaning that this must be a bacterium. So we have our answers: a plant cell in the case of figure 1, and a bacterium in the case of figure 2. In part B, we're asked a very simple question: why was an electron microscope used to create these images? Well, for the 1 mark, all you need to say is that an electron microscope has higher resolution, resolution being the key word, relative to the naked eye or to a light microscope. So, what is resolution? So, if we were to look at this image, a higher resolution image would be able to discern that these consist of 2 individual points. So, for instance, if we only had a light microscope, the naked eye would only be able to see this. Relative to a higher resolution image, with an electron microscope, we might be able to see that these are 2 individual points, separated. So our answer is that there is a high resolution in electron microscopes. Question C is a calculation question, where we're told that the scale bar below each image is 3 centimeters, and given that, what is the magnification of each image? Okay, so all we need to do is work out the magnification, which is done with a simple equation. Magnification is image size, which is 3 centimeters, over the real size, which in each scale bar is either 20 micrometers or 0.5 micrometers. So, we should bear in mind we have different units here, so our 3 centimeters and micrometers have to be equilibrated somehow, so we'll convert our centimeters into micrometers. In 1 centimeter, there is 10 millimeters, and there are 1,000 micrometers in a millimeter, ergo there are 10,000 micrometers in 10 millimeters. So, let's use our magnification equation, m equals I over r for simplicity. Our image is 3 centimeters, therefore in micrometers we would have 30,000. That has to be divided by both 20 and 0.5. Let's start with 20. Over 20 micrometers we have 1500 as our magnification, and we'll do the same for our 0.5 micrometers, and we'll have 60,000 as our magnification. So our answer to part 1 is that we have a 1500 magnification and also a 60,000 magnification to see figure 2, which was a bacterium, which is much smaller than a eukaryotic cell, which was figure 1. In question D, we're asked to discuss the evolutionary order of appearance of the mitochondrion, chloroplast, and ribosome, and to explain our reasoning. So it's not enough to just recall the historical fact. If you're familiar with it, we have to explain a rational basis for how we could come to that perspective. So let's have a think about the difference between eukaryotic structures and prokaryotic. Just as a reminder, eukaryotes are animal and plant cells, prokaryotes are bacterial cells for the purposes of this discussion. So we're asked to think about mitochondria, chloroplasts, and ribosomes. So we know that ribosomes are shared between prokaryotes and eukaryotes alike. So that means that ribosomes as a structure must have predated the divergence of eukaryotes from prokaryotes along the evolutionary chain. We also know that mitochondria only exist in eukaryotes, and the same can be said for chloroplasts, but those only exist in plant cells. So we can have a separate subset within our Venn diagram of plants. Given that mitochondria are in both plants and animals, they must have predated the divergence within eukaryotes into animals and plant cells, therefore our order must be ribosomes, followed by mitochondria and chloroplasts. So we can say our answer is that ribosomes were followed by mitochondria, which were in turn followed by chloroplasts along the evolutionary chain. Part E is another calculation question, where we're asked to work out the estimated percentage of the volume of the cell which the nucleus occupies in figure 1. So, we're given the equation 4 over 3 pi r cubed, which gives us the volume of a sphere, which we can take the nucleus to be, and we can take the volume of the cell itself to be a cube, according to the question. So we have an unknown value that we need to find, And we can remind ourselves that the radius of a circle, or indeed a sphere, is this value here, taken in a cross-section of a sphere, and using the scale in the question, we can find this to be roughly 15 micrometers in the case of our nucleus. So we can plug this into our equation and find the volume of the entire nucleus to be 14,137 micrometers cubed. So we can estimate the length of one side of our cube to be, namely our cell, to be 50 micrometers, which we can cube in order to find the total volume of the cell to be 125,000 micrometers, and doing a simple percentage calculation, this is 9.42% occupied by the nucleus. So that's our answer, although the examiners would have accepted between 6 and 20% as a final answer. In the final part of the question, part F, we're asked to discuss, in a 10-mark question, how the differences in the structure of the cells, shown in Figure 1 and Figure 2, namely a plant cell and a bacterial cell, affect the locations, so how the structures affect the locations of different processes within the cells. So, the best thing to do with these long-form questions is not to rush into it, writing everything you know, but to approach it systematically. We're asked about how processes are affected by the structures, which in turn affect the locations in which these processes occur. So what are the processes that happen within cells? So let's think about some. Well, the principal ones are respiration for energy, and there is also the cytoplasmic component of respiration, which is glycolysis. Bacteria can only do glycolysis, whereas eukaryotes, such as animals and plants, can also undergo mitochondrial respiration. There's also photosynthesis, since we're talking about plants in Figure 1, and there's also DNA replication, protein synthesis, cell division, and any others which perhaps relate to one or the other of bacterial or plant cells, which we'll think about later. So, in order to determine how these processes are affected in their location by differing structures between plants and animals, it's best to think about this in a table. So, what we'll do is discuss how the locations change between eukaryotes and bacteria, and why this is affected by structure. Well, the principal reason is that eukaryotes happen to have membrane-bound organelles. This enables compartmentalization, that membrane-bound organelles permit a greater surface area for reactions, whereas compartmentalization enables different chemical environments that exist within the cell for more efficient processes. So, in the case of eukaryotes, we have the mitochondrial membrane, where efficient respiration can occur, whereas the bacteria only have the cell membrane. We have glycolysis within the cytoplasm of both eukaryotes and bacteria. Photosynthesis occurs in the chloroplasts in plant cells, whereas the bacteria, such as cyanobacteria, can only carry out photosynthesis in their cell membrane. DNA replication occurs in the nucleus within eukaryotes, whereas bacteria have to rely on the cytoplasm as they have no true nucleus. Protein synthesis, which is integral to both, occurs in the ribosomes in both cases. However, in bacteria, these are cytoplasmic, whereas in eukaryotes, most of the protein synthesis occurs bound to the endoplasmic reticulum, or ER. Cell division, this involves mitosis and meiosis, which both involve the nucleus, for DNA replication, whereas the bacteria only have binary fission. Any other processes include lipid synthesis, which involves the Golgi apparatus, and hydrolytic enzyme synthesis, which involves the peroxisomes and the lysosomes, which only happens in eukaryotes. So, now that we've looked at the different locations affected by the different structure, which in turn affect different processes, now we should think about the marking scheme and how examiners would go about giving the 10 marks involved in this question. Well, there are a total of 12 marks up for grabs. The first mark available would be for noting that respiration occurs in the mitochondrial membrane, aerobic respiration that is, for eukaryotes. Chloroplasts are involved in photosynthesis in eukaryotes, that's the second mark. The third mark is for mentioning DNA replication involved in the nucleus. The next mark is available for mentioning that respiration and photosynthesis depend on the external membrane of bacteria. DNA replication occurs in the cytoplasm as there's no true nucleus, that's the next mark. Another mark is available for talking about glycolysis, the cytoplasmic component, the anaerobic component of respiration. The next mark is available for identifying that there is a greater surface area for reactions owing to the compartmentalized membrane-bound organelles, which is the next mark. The next mark available is talking about the different microenvironments which are generated by this process of compartmentalization, such as different chemical gradients or the availability of unique proteins. Moving on, the next mark available is for mentioning that there are some advanced processes which occur only in eukaryotes, such as ER-bound protein synthesis or meiosis in the nucleus. The next mark available is for mentioning any other cellular components, such as peroxisomes, lysosomes, the Golgi apparatus, or the endoplasmic reticulum, which are only present in eukaryotes. And the final 12th mark is available for doing what we should have been doing throughout the entire process, which is giving a direct comparison between eukaryotes and bacteria.